atoms make up molecules, molecules make up cells, cells make up tissues, tissues make up organs, organs make up organ systems make up organisms. So this is our chemical or molecular level. Understand and be able to put these in order if I present those on a test to you. That's a hint. Be able to go from the simplest to the most complex. The most complex is your organism. Everything else builds up to that. Okay? So we have the atoms, the cellular level, and they build the tissues. There's 4 types of tissues in the human body, and that's it. There's epithelial tissue, there's connective tissue, there's muscle tissue, and there's neural tissue. Okay? So the human body has 4 types of tissues that make up all organs. Organs make up the organ system. There's 11 organ systems in the human body. We went through each of those on Monday, and we'll go into detail in each one of those as the other chapters as we go. Come on. Okay. This chapter concentrates on cells and tissues. There's over 75 trillion cells in the human body. There are approximately 200 types of cells, all made up of the 4 different tissues. So all these cells can be placed in one of the 4 tissues, like we said, epithelial, connective, muscle, or neural. Epithelial tissue characteristics. The cellularity, the cells are bound close together. Epithelial tissue makes up your skin, and all those cells are bound tightly together. There's no intercellular space. The cells match up and line up with one another. Epithelial tissue has polarity, which means there's one end and another end, and they're extremely different. The exposed end, or the top end, is your apical surface. The surface that attached to the connective tissue underneath is the basal surface. So there's a polarity with epithelial tissue. There's an apical surface and a basal surface. Surfaces are structurally and functionally different. So polarity is reference to this structural and functional difference. That's what we call it a polar cell. Okay, so you can see the apical surface of the cells have cilia or microvilli. This is the apical surface. That's where your absorption, your secretion, everything else occurs. That's the apical surface. You can see how these cells are fused together, held together with tight junctions. Okay, the tight junctions prevent movement of pathogens or diffusion of dissolved materials between the cells. Folds of the plasma near the base of the cell membrane increase the surface area to expose the plasma lemma. Okay, so the plasma lemma here, okay, is adhered together. At the basal part, there's folds into the plasma lemma, which increases surface area. Increases surface area allows for a greater binding between the basal part of the cells to the plasma, from the plasma lemma to the basal portion of the cells. Okay, so it basically increases your surface area. And you can see the Golgi apparatus, the nucleus, mitochondria, etc. Okay. Epithelial attachment, the basal layer is attached to the basal lamina. Okay. Epithelial tissue does not contain blood vessels. So there's no blood vessels in epithelial tissue. They're arranged in sheets composed of 1 or more layers of cells. Their regeneration ability, cells are continuously replaced by mitosis. Those cells undergo constant reproduction in epithelial tissue. That's not the same for neural tissue or muscular tissue. So epithelial undergoes constant regeneration and replacement through mitosis, which we talked about. The function of epithelial tissue provides physical protection, protects from abrasion, protects from dehydration, and prevents from destruction. Okay, and it prevents or protects the structures underneath. Okay, it controls permeability, so water cannot get in or out, allows you not to dehydrate, and it makes the body waterproof. So all the water in the surrounding environment doesn't enter your body. Provides sensations. Say when it's windy, you can feel it. When it's cold, you know it. Provides stimuli from the external environment into the nervous system. And it produces secretions. Remember we talked about the hair follicles have sebaceous glands. They produce a mucus or oily substance. Your skin has sweat glands that produce a watery substance. Okay? So epithelial controls permeability, sensations, and the secretions. Okay, we'll give you the different examples of each of those. And click. Okay. Epithelial cells have microvilli, stereocilia, or ciliated epithelium. The microvilli is the very small. They increase the surface area, and they increase the surface area to increase absorption and secretion. Microvilli are always found on the apical surface of cells of urinary and digestive tract. That's where you absorb, that's where you secrete. Okay, it's always on the apical surface, and they increase surface areas. That's a really good true-false question for a test. Okay, microvilli are found on the apical portions of epithelial cells. Stereocilia are long microvilli. Okay, they're found in the inner ear, male reproductive tract. Ciliated epithelium moves substance over the apical surface, and they're found in the respiratory tract. That's your ciliated epithelium. Remember we talked about that, where they beat in one direction. They constantly move pathogens, everything else in the mucus on out. And here's a picture of what they look like. This is the microvilli. These are the real small ones right here. Okay, they increase surface area, allow secretions and absorption. The cilia are a lot longer. These usually beat in one direction to move things, like in the lungs. They beat upward. So they take, like I said, pathogens and down, and they move it up with mucus, and that's when you cough it out of your system. So things don't go deep into your lung tissues. Okay? And again, you see the cilia and the microvilli on the apical surface of the cell, not on the basal surface. They always occur on the apical surface. So the apical surface frequently bears microvilli. A single cell typically has only one type of process. Tight junctions, these are the junctions right here between the cells, hold those together. Okay, the folds of the plasma lemma right down here, those allow an increased surface area to the basal lemma to help increase the attachment that you have between the cells and the basal lemma to help hold that together. Okay, maintain the integrity of epithelium. 3 factors are involved in maintenance of the integrity of the epithelium. You have intercellular connections, you have attachments to the basal lamina, and your epithelial maintenance and renewal is self-perpetuating. In other words, it continues to go regardless of anything. It's had a constant signal to regenerate, and it's going to constantly replace itself. The intercellular connections, we talked about those before. Extensive connections between the cells holds the cells together. Okay, if you had your skin, your epithelial covering of your skin, if there are a lot of holes in there, what would happen if you went for a swim? All that water would go into your body. Okay, so it's tight contacts between there to hold the cells together. Prevents the passage of chemicals and pathogens. If bacteria lands on your skin, it prevents it from entering. It's the first line of defense that your body has. Okay, the cell junction, we use the cams. Remember, those are like the little zipper molecules that line up and they hold it and interlock. Those are cell adhesion molecules, and the intercellular cement holds the epithelial strength and stability. The proteoglycans are your intercellular cement that helps hold together along with the cellular adhesion molecules. Help holds all that skin together. And this is how the epithelium is packed together. You can see on the slide here, you can see that all these layers right here, as they're following the mounds, those are your cellular adhesions that keeps the skin intact and keeps everything together. Now it attaches to the basal lamina. Remember, it goes in and out and forms invaginations to help increase its surface area. So the plasma lemma attaches to the basal lamina. The plasma is this at the basal surface of the cell. Remember the apical and you have the basal surface. There's 2 layers when it connects to the basal lamina. There's a clear layer and there's a dense layer. So the basal lamina attaches to the underlying connective tissue, and that's what holds the cell to the body. And here you can see the 2 layers. So this is your cell. These are your CAMs, your cell adhesion molecules, and your proteoglycans, your intercellular cement. So this junction is held together very tightly, so things cannot pass. On the plasma lemma, you got the imaginations, and there's 2 layers that hold to the connective tissue. There's a clear layer, and there's a dense layer with more fibers in it. And that's what holds the cells to the connective tissue below it. Okay. So the epithelium must be replaced frequently. Why? Because it's constantly exposed to irritants. It's constantly exposed to the environment. Okay, it's constantly being eroded against, and it has to be renewed to protect itself. So you must replace it frequently because of exposure to disruptive enzymes, which you find in the environment, toxic chemicals, smog, cigarette smoke, all kinds of toxic chemicals your body's exposed to, pathogens, bacteria is everywhere. Okay, it's on your skin, mechanical abrasion. Okay? It protects the skin from having lacerations through it and losing the internal portion of your body. It's replaced continue with time, and it compounds the division of stem cells. Stem cells replicate, they become more specialized, and they start replacing different epithelial cells. Epithelial cells has two classifications, which you need to know for my class and for your nursing exams. Two classes of epithelium, simple and stratified. Okay, epithelium has only one layer of cells. Okay, stratified has two or more layers. That's the big difference. Okay, simple epithelium is one layer of cells. Stratified epithelium is two or more layers of cells. Okay, in simple, we have epithelium has one layer, nuclei are approximately the same level within each cell. Okay, it's found in protected areas such as the internal compartments of the body. You wouldn't have simple epithelium in areas that are exposed to trauma because we wear through the layer way too fast and you lose your protection. Simple is where it's already protected inside the gut for absorption, inside the lungs for passage of airway, of oxygen, CO2, things like that, where it's protected by the body. And you need a thin layer for a diffusion to occur. That's where simple is. Stratified, found in areas where there are mechanical or chemical stresses. That's where your stratified epithelium would be. The outside of your skin, your digestive tract, where your food's going in, your esophagus, where you're getting trauma all the time, that's there to protect it. Okay. Epithelial tissues, you have different types of cells. And this will have to identify these histologically, which we'll do in the lab. We'll show you different histological slides. Okay, you have squamous cells. Squamous cells are very easy. They're flat. Okay, they're thin, flat, squished nuclei. Very flat cells. Cuboidal cells are shaped like a cube. They have a centered round nucleus. Okay, so the length and the width are roughly the same. They're the shape of a cube. Columnar cells are shaped in a column. They're longer than they are wide. Okay, nucleus is near the base. Transitional cells. Transitional cells is a mixture of cells. The nuclei appear to be scattered. So you got squamous cells, cuboidal cells, and columnar cells, and then transitional cells. And each of those occur in different areas of the body, depending on the different functions that they're going to be serving. So simple. Remember, simple was one layer. They're very delicate cells. Location is lining body cavities, like the heart, blood vessels, areas where it's protected. Okay? They reduce friction. They absorb and secrete materials. Again, they can absorb materials and secrete because there's only one layer, and they're protected so they're not getting the abrasion and the trauma as other parts of the body are. Okay, this is what they look like. It's a single layer. Again, the mesothelial ventral body conduit, the endothelial lining of the heart and blood vessels, where diffusion has to occur. Okay, it reduces friction, controls vessel permeability, and performs absorption and secretion. This is a histological slide, H&E stain, of a simple squamous epithelium. Simple means one layer. Squamous means flat, squished cell. So if I showed you that slide, you'd be able to tell me it's simple squamous epithelium. Okay? Again, it's simple because there's only one layer. We see the layer right here. It's squamous because it's flat, and it's epithelial tissue. Okay? Stratified squamous epithelium. Stratified means what?
Two layers.
Two or more layers, perfect. Okay. Location is the surface of the skin. We have multiple layers, and it's squished so it can adhere to itself, and it protects. Lines the mouth, esophagus, anus, and the vagina. All areas where you can get trauma into those areas. Okay. It has to protect those areas, so it's stratified squamous epithelium. protects against abrasion, pathogens, and chemicals. And you can see the difference. Look at how thick this area is. This is at least 1, 2, 3, 4, 5, 6 layers. And you can see as the cells mature, as you get closer to the superficial surface here, they become flat, squished cells. A flat, squished cell is squamous cell type, okay? And it provides protection. And it's stratified because there's 2 or more layers, squamous because it's flat, and it's epithelial. Okay? That's where we're going to start to name things. Simple, cuboidal epithelium. Again, what is, you'll be able to tell what that looks like, right? Simple means 1 layer. Cuboidal means it's shaped like a cube. And epithelial tissue. And thyroid gland, ducts, kidney tubules, secretion and absorption. Anytime you have simple epithelium, it's going to be because of secretion or absorption. It's easy to transfer between only 1 cell. It's a lot harder to penetrate more than 1 cell. Okay? And secretion and absorption is their function, very, very limited protection. You don't get much protection because there's only 1 layer. Okay? This is an example of a kidney tubule. Okay? You see it's simple because it's 1 layer. You see it's cuboidal because they're shaped like a cube. Okay, everybody see that right here in this little box. You can see here it's on in the histological slide, which is something that you see on your practicum. You can see you have 1 layer, and you can see it is in cube shape. So simple, cuboidal epithelium. Okay, stratified cuboidal epithelium. Stratified means 2 or more layers. Cuboidal means the shape of a cube. This type is really very rare. It's a duct of sweat glands, and it is for secretion and absorption. Okay, lining of some ducts for sweat glands, and again, you can see here we have 2 layers. Here's an inner layer around this lumen, and there's another layer around it. So that's where the word stratify comes from. You have 2 or more layers, and you see the shape of these cells. They're not flat, they're in the shape of a cube. So stratified, cuboidal epithelium. Simple columnar epithelium. Again, simple 1 layer, taller than they are wide because in shape of a column, epithelium. They line the stomach, intestines, gallbladder, uterine tubes, and collecting next to the kidney. They function as secretion, absorption, and some protection. Okay, not a lot because they are simple, only 1 layer. So you have simple columnar epithelium. See how the shapes are columnar. They're very tall and they're narrow. So they're in the shape of a column. Okay. To get more specific with this, if I showed you this slide on a test, you'd get it wrong if you put simple columnar epithelium. This is ciliated. This layer right here, this is above the cell. This is cilia. So the actual definition of this, which we'll get into a little later, is ciliated, simple columnar epithelium. You have to have the detail down. Okay? You have to know exactly what you're looking at. So this is ciliated. This is simple, 1 layer, columnar epithelium.
Question.
Everybody sees what the cilia looks like on that. So you put ciliated, simple, columnar epithelium.
Are simple and stratified always found together, or are they always, or can they be separate?
They have to be separate because if there's more than 1 layer, it becomes stratified. If it's only 1 layer, it's simple. If there's more than 1 layer in that sample of tissue, then it's stratified. So it would be one or the other. They can't be mixed together. Does that make sense?
Yeah.
Okay, perfect. Because simple is the 1 layer. If it's only 1 layer of cells, it's simple. If there's more than 1 layer in the simple in the sample you're looking at, then it's stratified. Now, can one place in the body have both simple and stratified? Yes. But it depends. The sample of the tissue I'll give you will be one or the other. Okay. So stratified columnar epithelium is located in the pharynx, epiglottis, the anus, mammary glands, salivary glands, and urethra, and it's for protection. Whenever you have stratified, a lot of times that's for protection because there's multiple layers. And you can see here, you see where the lumen is. Okay. And you see going away from the lumen, you have several layers. There's 1, 2, 3 layers. You can see it better out here where it's drawn out. Okay. But these are your columnar cells, and these are the multiple layers where they're coming from. in the deeper basilar region. And this is where they're connected to the connective tissue where they're attached. Okay, so again, this is stratified columnar epithelium. And this is ciliated. You can see the ciliated layer right here. It doesn't show it on this one here, but you can see the little cilia on the edge right here. So it's ciliated, stratified columnar epithelium. And again, when it's stratified like that, stratified is for protection from abrasions, from other irritants, from pathogens. It helps provide a layer of protection. Pseudostratified, ciliated columnar epithelium. Pseudostratified means it kind of looks like it. Okay, means that it looks like columnar epithelium. The nuclei are situated at different levels: nasal cavity, trachea, bronchi, and they function as protection and secretion. And you'll see here, you got kind of a mixture of cells. Most of it looks like it's columnar, so it's called pseudostratified because there's more than one layer. Okay, ciliated because you can see the cilia on the top here. Columnar because those are basically columnar here. You got some different shapes that could almost be considered cuboidal. Okay, epithelium. So it's what's called pseudostratified. Okay, because it's going to look like either one. Transitional epithelium occurs in the renal system, urinary system. Basically, the urinary bladder consists of many layers. It's a combination of cuboidal and oddly shaped cells. Because what it's going to do is stretch. Okay, one function of the urinary bladder is when it's empty, it will have a certain shape. When it fills, those cells have to stretch. Okay? As they stretch, that's transitional epithelium. The cells transition to a different state, which allows the epithelium the ability to stretch. Okay? When you look at the urinary bladder, you'll see transitional epithelium. Okay? So you see you got some funky different shapes of cells here, especially when you look at the light section over here, light microscope section. Okay? This is transitional, different types of cells inside of there. And again, this is in the relaxed bladder. Now, when it starts to stretch, these cells start to become flat. Okay, so you might guess squamous, but they're not. They're transitional because when it goes back again, it goes back to this almost cuboidal shape. So this is transitional epithelium. One place you're going to find it is in the urinary bladder because it has the ability to stretch. As it stretches, it changes shape. All right, glandular epithelium. We have glands in epithelial tissue. Many epithelial tissue contain gland cells. Glands are classified based on the type of secretion they release, the structure of the gland, how it's physiologically shaped, and the mode of secretion, what type of secretion it undergoes, how it releases its products into the target area. You have types of glands. You have exocrine glands and you have endocrine glands. Exocrine glands release its secretions outside the body. Okay, sweat. Sebaceous glands for your hair cells. Those are exocrine glands. They release it outside of the system. Endocrine, release it into the system. Release glands into your bloodstream, into your lymph system. Okay, those are your hormones, things like that. So exocrine releases its secretions outside. Endocrine releases its secretions inside, like your blood vessels or your lymphatic system. Okay. We have the different types. We'll go over the different types of them here too. Okay, so exocrine glands, secretions travel through ducts to the epithelial surface. Endocrine, secretions enter into the blood or the lymph. Okay, when they enter the blood or lymph, they're termed hormones. So exocrine gland categories. Exocrine glands go to the surface of the skin, and they can be serous glands, secrete watery fluids rich in enzymes. They can be mucus glands, secrete glycoproteins, mucins, that absorb water to produce mucus. You have mixed exocrine glands, contain both serous and mucus secretion. So you have serous glands, mucus glands, and you have mixed exocrine glands that can secrete both. Endocrine glands release their secretions by exocytosis. Remember, exocytosis is an active transport requiring ATP. Remember, the secretory vesicles break off from the Golgi apparatus. They travel along the microtubules to the plasmalemma. They fuse with the plasmalemma, and then they open up, okay, and they release their product into the bloodstream or into the lymph. Okay? Those are called hormones released into the body. So those are your endocrine glands. That's the difference between exocrine and endocrine glands. The gland structure can be unicellular, which is one cell, and they secrete mucins. There's two types. There's goblet cells and there's mucus cells. Or it could be multicellular, where you secrete mucins and it produces secretory sheets of cells as its byproduct. For the unicellular cells, goblet cells found among columnar epithelium of the small and large intestine. That's where they release their products. Mucus cells found among pseudostratified ciliated columnar epithelium of your trachea. Okay, trachea is your windpipe here and the stuff that you cough up. When bacteria, pathogens, anything in the air that you breathe, that mucus, that mucus being produced traps those pathogens. So it doesn't travel down deep into your lungs. That's why it's produced in the trachea. Then the cilia beat upward and beat that material out of your system. And it comes out and you cough it out. Okay? And again, the cilia in the trachea beat in one direction only. They beat outward. So they take all that mucus that they're producing, traps the pathogens, and beats it outwards. Multicellular produce exocrine secretions. They consist of a portion that produces the secretion and a portion that carries the secretion to the epithelial surface. Okay, so the exocrine secretions have two sections. The other ones produce endocrine secretions. Okay, multicellular glands can be tubular, alveolar, or tubulo-alveolar. In the tubular, the cells form tubes. In the alveolar, the cells form blind pockets. In the tubulo-alveolar, they form both. They form tubes and they form blind pockets. I'll give you some examples in just a second. You can also have simple and compound. Simple do not have any branching ducts. It's a simple tube. Simple tubular is found in the intestines. Simple coiled tubular is found in the American sweat glands. Simple branch tubular is found in gastric glands and mucous glands of esophagus, tongue, and duodenum. I might sound contradictory, simple and branch, but it's each individual tube, but then they connect together so we form a branch system. But each tube does not have branches off of it. Okay, simple alveolar, which is found only during development stages of glands in the developing fetus, and simple branched alveolar, which is found in your sebaceous glands. Here's some examples. If you look at this, you can see simple glands. Simple tubular, you can see one tube. Okay, simple coiled tubular, it's one tube and now it's coiled. Simple branch tubular, it comes down here and there's branches that they fuse together, but each of these does not branch. That's why it's simple. So it's a simple branch tubular. Alveolar has a round pocket now. It just doesn't dead end in an end, but it has a bulbous extension like an alveoli. That's what's called alveolar gland. So this is simple alveolar. This is simple branched alveolar. Okay. Simple and compound. Compound glands have various branching ducts. So the simple has no branches. Compound have branches. Compound tubular is found in mucous glands, both the bulbourethral gland and testes of the male sex system. Compound alveolar is found in mammary glands, and compound tubulo-alveolar is found in salivary glands, respiratory tubes, and in the pancreas. And here's your examples. These are compound. You see how each branch branches. That's the branch system. So this is a compound tubular. You see where the tube is advanced. You just come in and they end in a tube ending. Just a straight ending, no swelling at the end of it. And these are alveolar. You can see how they swell at each of these, and they add like an alveolus in the lung. Okay, this is a compound alveolar gland, and this is a compound tubulo-alveolar gland, where you have some tubule ones, like this is a tubule, this is tubule, this one's alveolar. This is the way that it's shaped. Okay, so you have compound tubular, compound alveolar, and compound tubulo-alveolar. Those three types of compound glands. Okay, we have different types of secretions. You need to know these for your nursing exam and for my exam. Okay, they're going to secrete. There's three different ways a gland can produce its secretions, whether it's to the outside or whether it's to the blood inside to the bloodstream or the lymphatic system. There's merocrine secretion, there's apocrine secretion, and there's holocrine secretion. Okay, those three types. Merocrine secretion is exocytosis, and it's found in salivary glands. Exocytosis is secretory vesicles break off from the Golgi apparatus, travel on the microtubules, come to the plasmalemma, fuses with the plasma lemma, opens up, and allows it to come on. Exocytosis is a active transport, requires ATP. Apocrine secretion. The apocrine portion or the apical portion of the cell blows apart. So basically the apical third of the shell shreds apart and it releases all the vesicles that are in there. Okay, those are your mammary glands. That's how milk is released into the tubules for infants. Holocrine secretion. Holocrine secretion, the entire cell bursts apart. The entire cell is filled with all these little vesicles. And then the whole cell blows apart and all these vesicles are released into either into the internal or external surfaces. Okay, those are found in sebaceous gland. So merocrine secretion, apocrine secretion, and holocrine secretion. This is merocrine secretion. Merocrine secretion is your salivary glands. And you can see the picture here. These are your secretory vesicles. These are released from the Golgi apparatus. Okay, and they migrate along the microtubules. See, here's your Golgi apparatus, little pancake thing. This is a trans. This is a cis end where the vesicles are released. They migrate along microtubules. They reach the plasma lemma. They reach the plasma lemma. They fuse with it, and they release their products. Okay, that's merocrine secretion. Apocrine secretion, the apical or the top 1/3 of the cell. So here's your entire cell. The top 1/3, the apical portion. Remember that these cells are polar. There's an apical, there's a basal. The basal cell is attached to the basal lamina. The apical cell has your alveoli for all that for secretions and absorptions. Okay, so the top 1/3, the apical portion breaks apart and releases all the secretory vesicles. Okay, that's apocrine secretion. Holocrine secretion, the entire cell blows up. The entire cell destroys itself and all these little vesicles here get released. Okay, so the whole cell blows up. So merocrine, apocrine, and holocrine secretion are the three different types. All connective tissues have three main components. They have specialized cells. Remember, connective tissue and testicles that holds all these epithelial cells into the body. Epithelium is this outside lining. It's all connected to and supported by this connective tissue at the base of the layer. So epithelial cells are polar. The apical portion is outside. The basal portion connects to connective tissue. That's what holds everything together in your body. So all connective tissue that we're talking about is going to have three components: specialized cells, extracellular protein fibers, and a matrix. Matrix is a term for all these extracellular components of any connective tissue that's made of protein fibers and what's called ground substance. So it's this mesh that holds it together inside the connective tissue. Okay. So you're always going to have this protein fibers and ground substance inside this matrix holding everything together. And that's this is occurring at the basal layer of epithelial cells. Functions of connective tissue. This is an exam question. Okay. The function of connective tissue: establishing the structural framework of the body, transporting fluids and dissolved materials, protecting organs, supporting, surrounding, and connecting other tissues, storing energy, and defending the body from microorganisms and different pathogens. Classification of connective tissue: you have connective tissue proper, you have fluid connective tissue, and you have supporting connective tissue. So connective tissue proper is a matrix of fibers, those loose fibers and dense we talked about that hold things together. The fluid connective tissue is a matrix of liquid. Your blood is connective tissue. It's fluid connective tissue. It's blood and lymph are your two types of fluid connective tissue. Supporting connective tissue has a matrix consisting of gel or solid, like cartilage or bone. Those are supporting connective tissues. So blood is a fluid connective tissue, and bone and cartilage are your supporting connective tissues. So here's a great diagram that shows it right here. This is connective tissue. You have connective tissue proper, you have loose, and you have dense. Okay, this creates loose open framework. This is areolar tissue, adipose tissue, and reticular tissue. The dense connective tissue proper, they're packed together tighter. It's more dense. There's dense regular, dense irregular, and elastic. Fluid connective tissue is blood, cardiovascular system, and lymph, okay, which is fluid containing the lymphatic system. Supporting connective tissue, what's called support, is the cartilage in your bone. Cartilage is a solid rubbly matrix. You have hyaline cartilage, you have elastic cartilage, and we have fibrous cartilage. And bone. Bone is a structure that holds the entire body together, gives you support. It's solid, crystalline matrix. Okay, that's a great diagram there that shows you the different types of connective tissue, the 3 types. Okay, connective tissue proper. This is the first one we talked about. There's 2 classes of connective tissue proper. There's fixed cells and there's wandering cells. As you can guess, fixed cells stay in one place in the connective tissue. The wandering cells wander throughout the body, going where they're needed. So fixed cells are your mesenchymal cells, your fibroblasts, your fibrocytes, your fixed macrophages, your adipocytes, and your melanocytes. Okay, those are the different types. And again, this tells you what the different types are, the fixed cells. Fibroblasts produce connective tissue fibers. Fibrocytes maintain connective tissue fibers once they're produced. Fixed macrophages phagocytize pathogens and damaged cell. They eat and dissolve them, like the lysosomes and the peroxisomes. Okay? They phagocytize, which just means dissolve pathogens and damaged cells. Adipocytes is your fat storage, your lipids. Mesenchymal cells are stem cells for your connective tissue. They differentiate into the different cell types needed in the connective tissue. And melanocytes synthesize melanin, which controls your skin color, your pigmentation. Okay, connective tissue proper. There's 2 classes of connective tissue proper cells. There's fixed cells and the wandering cells. We talked about the fixed cells. Now the wandering cells. The wandering cells are your free macrophages, your mast cells, your lymphocytes, neutrophils, and eosinophils. Those are different types of white blood cells. And we'll go through the functions of each one of those as we go on. So free macrophages, mobile, they travel. They do the same thing as the fixed macrophages. They eat and they dissolve. They phagocytize thick pathogens, cell debris, things like that. But they're mobile. They go where they're needed. Mast cells, okay, they stimulate local inflammation. When you have trauma to an area, you get an inflammatory response. The first step is your mast cells. The lymphocytes participate in your immune response. Those are from the white blood cells. And your neutrophils and eosinophils are smaller phagocytic blood cells that mobilize during infections or tissue injuries to help destroy damaged or diseased tissue. Okay. The 3 types of fibers are associated with connective tissue. We talk about collagen fibers, reticular fibers, and elastic fibers. All right. So you can see the 3 different types on the diagram right there. The very thin, small reticular fibers. The elastic fibers are very flexible, and the collagen fibers are a lot thicker, supporting your main structure. Okay. The connective tissue has loose fibers and dense fibers. The loose fibers are your areolar tissue, adipose tissue, and reticular tissue. Your dense fibers are your dense regular, dense irregular, and elastic. Okay. The areolar tissue, the first type, okay, is deep dermis. You got your epidermis, you got your dermis, and the deep part of the dermis is where your areolar tissue is. It's between muscles, around blood vessels, and around the nerves. That's where it's located. Connects the skin to the muscle. Provides minimal support, but is independent of movement. Allows your skin to slide and move without the muscle moving underneath it. And the matrix that it is involved with is the different fibers. Okay, you can see the areolar connective tissue of the pleural cavity. You can see the fibrocytes, macrophages, collagen fibers, which are thicker fibers, mast cells produce your inflammatory response, and elastic fibers to give it flexibility. Okay. Adipose tissue. Adipose tissue is in the hypodermis. So you got your epidermis, your dermis, and your hypodermis. Okay. So the adipose tissue, your fat cells, are in the hypodermis, in your buttocks and surrounding organs. The function is for cushion and insulation. And the matrix again for this is also fibers. Okay, these are your adipocytes. This is your white adipose cells. Okay, that's fat storage, which is stored for energy. The reticular tissue. Reticular tissue is your liver, spleen, kidney, lymph nodes, tonsils, appendix, and bone marrow. Functions as a supporting framework. Okay, and again, the matrix of this is also fibers. Okay, it's found in the liver, kidney, spleen, lymph nodes, and bone marrow. And you can see all the reticular fibers inside this picture from the liver. Okay. You also have dense regular connective tissue. Location is your tendons, aponeuroses, ligaments, and elastic tissues. Okay, tendons connect muscle to bone. Aponeuroses connect muscle to muscle. Ligaments connect bone to bone. Elastic stabilizes the vertebrae. And the matrix is fibers. You need to know those four. You need to know tendons, aponeuroses, ligaments, and elastic, and what connects to what. Okay? You can see the different collagen fibers in it, and then the fibrosite of the nuclei. Remember, the fibroblasts produce the collagen fibers. The fibrocytes maintain collagen fibers. Okay, elastic tissues are found in the vertebrae, between vertebrae, what's called a cartilaginous pads, which we'll study as we go along. All right, and now we got dense irregular connective tissue, and that's found in nerve and muscle sheaths. Sheaths that go around and protect or encompass or encircle the nerve and the muscle, and these provide strength. Okay. We see that collagen fibers are thick fibers, and they provide strength to the muscle and nerve tissues. Okay, fluid connective tissue, two types, blood and lymph. Blood has erythrocytes, which are your red blood cells, leukocytes, which are your white blood cells, platelets, which are portions or clumps broken off from what's called a megakaryocyte that circulate in blood and plug holes, okay, prevent blood loss, and plasma, which is the liquid portion of your blood. Okay, so locations are towards erythrocytes, red blood cells, transport oxygen and carbon dioxide. Now, everybody remembers that red blood cells transport oxygen. So on your nursing exams, they have a question on there. They'll have it every time in the end class. It says circle everything that is transported by the red blood cells. You circle both oxygen and carbon dioxide. Now, oxygen and carbon dioxide, oxygen is transmitted by red blood cells from the lungs to the cells. Carbon dioxide is transported by red blood cells from the cells to the lungs. The carbon dioxide is also transported free in the bloodstream. Okay, but it also binds to red blood cells. Now, carbon dioxide does not bind to the same binding site as oxygen does. So it does not inhibit in any way the transportation of oxygen. It's a completely different binding site. It does not bind with hemoglobin. Leukocytes, your white blood cells fight infection. Platelets are for your blood clotting. And your plasma is your liquid portion of the fluid connective tissue matrix. Red blood cells, white blood cells, there's 5 different types, and we'll go through each of those. You got monocytes, lymphocytes, eosinophils, neutrophils, and basophils. And we'll go through those in greater detail. And then platelets go around and they cause clotting of blood from a traumatic injury. Lymphatic system, you have lymphocytes. Lymphocytes develop into T and B cells, okay? And they're involved with your immune response. Supporting connective tissues, cartilage and bone are supporting connective tissue. And there's a difference between the two. Cartilage is the gel matrix made up of chondroitin and sulfate. The cells reside in lacunae. Cartilage is a lot more flexible than bone. Bone is a solid matrix made of calcium phosphate. And the cells reside in the lacunae also. So both cartilage and bone share a common feature. They both have lacunae. Lacunae is a indentation or a little space in the bone where the fibroblasts live or where the osteoblasts live. And those produce either bone or cartilage. Okay? So lacunae is common in both cartilage and bone. There's different types of cartilage. There's hyaline cartilage, there's elastic cartilage, and there's fibrous cartilage. Hyaline cartilage, connection between your ribs and your sternum, connection with the joints of your elbow and your knee. Tracheal cartilage rings that keep you, remember I told you the trachea never collapses like the esophagus. The trachea stays open because you have these tracheal cartilage ring. That's hyaline cartilage. They're flexible support and they reduce friction, and their matrix is a gel. And you can see functions at your pectoral pectoral joint. You have chondrocytes. This is the matrix here, and the chondrocytes inside the lacunae. Those are the indentations or depressions in the cartilage itself. Elastic cartilage, the auricles of the ear, the epiglottis, the tube that folds over the trachea, so when you're eating or swallowing, it doesn't go down your trachea into your lungs. And the auditory tube. Auditory tube connects your middle ear into the pharynx in the back of your throat. Allows pressure to equalize in behind your eardrum, so they don't pop or burst. And they're also for flexible support. Okay, this is location. This is auricle of the ear. Provides support, tolerates distortion without damage, returns to its original shape. That's elastic cartilage. All right, fibrous cartilage. Pads of the knee joints, pads of the spinal vertebrae, the pubic symphysis. It resists compression and it absorbs shock. These are fibrous cartilage. Okay, you can see it in the spinal cord here. Okay, pads with the knee joints, pubic bones, and pelvic intervertebral discs. Again, you got your chondrocytes, and those are in lacunae, and then your fibrous matrix. Okay, bone. Bone is a lot harder, more brittle than cartilage. Location of bone is in your skeletal system. Okay, support and strength, and it is solid. It's made up of lamellae, which are different concentric rings. Now, each bone unit is called an osteon. Osteons consists of a central canal, osteocytes, the lacunae, which are the depressions, canaliculi, which are the very small, tiny canals, and the matrix of the lamellae. Okay, this is your osteon. This is a bone unit. Okay, inside you have blood vessels and nerves. So the artery, the vein, and the nerve usually all run together, okay, through the bone and innervate the different areas. You have compact bone on the outside, spongy bone on the inside. We will cover that. But each osteon, okay, has its own supply of blood from an artery and a vein. Okay, when you're, I don't know how many you give blocks, or when you aspirate, when you give a block to numb something, you aspirate on a syringe. You pull back on it to see if blood's there, because you don't want to go into the bloodstream if you want to just get into the fatty deposits. Okay, same when you're doing dentistry, you're giving a block for a nerve, you want it to go around the nerve. You do not want it getting into the blood vessel, because we have epinephrine there, and it causes the heart to palpitate and things like that. Okay, so blood vessels, arteries and veins and nerves all run together. That's why you aspirate, to make sure you're not injecting into the artery or the vein. Okay. So this tells you the difference between cartilage and bones. It's a real good summary. It tells you the cells, chondrocytes in lacunae, osteocytes in lacunae. They're both in lacunae, chondrocytes versus osteocytes. The matrix is chondroitin sulfate. Matrix of bone is calcium phosphate. Fibers are collagen, elastic, reticular fibers. And fibers in bone is the collagen fibers predominate, the bigger, thicker fibers. Vascularity, big important difference. Cartilage has no blood supply. Bone has an extensive blood supply. If you break a bone, ever treated a broken bone, it bleeds and it hurts. So nerves innervated and blood vessels innervated. Okay, so the vascularity of cartilage is nil. There is no blood vessels in cartilage. In bone, there's a lot of blood vessels. The outer layer of the cartilage is called the pericardium. The outer layer of bone is called the periosteum. The strength of cartilage is very limited. It bends easily, very hard to break, but it will bend. Bone does not bend very well at all. It resists distortion until it reaches a certain point when it snaps. Okay? So cartilage will not snap, it will bend. Bone will not bend, it will snap. There's two types of growth for cartilage, which we'll talk about. There's interstitial growth and there's appositional growth. Bone grows by appositional growth only. Okay, repair for cartilage. Repair for cartilage is very limited. Cartilage usually cannot be repaired. Bone, easily be repaired. We repair bone all the time. So cartilage has very limited ability to be repaired, whereas bone has extensive ability to be repaired. Oxygen demands. Oxygen demands of cartilage are low. Oxygen demands of bones are high. Think about it. Cartilage has no blood supply. You're not getting oxygen in there. The demands are low. Bone has an extensive blood supply. So you're going to get oxygen delivery there. It's very high. Nutrient delivery by diffusion through the matrix for cartilage because there are no blood vessels. For bone, it's by diffusion through the cytoplasm and fluid in the canaliculi as it gets delivered. All right, match tendons to which one? What are tendons?
Connects muscle to bone.
Connects muscle to bone? You got it. Perfect. Aponeuroses.
Muscle to muscle.
Perfect. Muscle to muscle. Ligaments.
Bone to bone.
Bone to bone.
Bone to bone. And elastics.
Stabilizes the vertebrae.
That one's tough. Yeah. All right. I guarantee you'll see a question like this on your midterm. Okay. So don't miss it. Got all four of them right there. Talk about membranes. All right. You have epithelial and connective tissue combined to form membranes. Each membrane consists of sheets of epithelial cells and underlying connective tissue. Okay, remember, connective tissue is always underlining the epithelium. That's what's formed the support. Connective tissue is not polar. It's a mass of fibers that hold it together. Epithelium is polar. It has an apical portion and a basal portion. Apical portion is the outside that extends to it. The basal portion is the part that connects to the connective tissue and holds the cells to the body. So there's four types of membranes that we have. There's mucous, there's serous, there's cutaneous, and there's synovial. And we'll go through each one of these. Mucous membranes. Mucous membranes, that's what you expect, produce mucus. They line your digestive tract, respiratory tract, reproductive tracts, and the urinary tracts. Okay? They form a barrier that resists pathogen entry. Okay? Whatever's trying to get in your body, the bacteria or the pathogen, is going to get stuck in the mucus. And it's not going to be able to penetrate. Okay? Keeps the epithelial surfaces moist, keeps us from drying out. Connection of the epithelium with underlying tissue is called the lamina propria. That's the connecting point between the epithelium and our connective tissue, is the lamina propria. Provides support for blood vessels and nerves that run through the mucous membrane. Remember, epithelium does not have blood vessels. The mucous membrane supplies the support for the blood vessels and nerves. Okay, you can see here, you can see the polarity of the epithelial cells. This is the apical, this is your cilia on it. Okay, this is the basal, this is the lamina propria, which connects the basal surface of the epithelial cells to the connective tissue. Blood vessels do not run in epithelium. Blood vessels run in the lamina propria. This is your arteries and your veins, okay? That's in your connective tissue. That supplies your nutrients to your epithelial layer. Makes sense? So on the test, if you see a question, true, false, that says blood vessels occur in the epithelial layer to supply nutrients to the developing cells. What would you put? False.
False.
Right? Perfect. So don't miss that one either. All right. Let me here. And you see the layer of mucus secretion over the microvilli there. All right, serous membranes. So those are mucous membranes. Serous membranes. Serous membranes line the body cavity, consists of parietal and visceral layers. Remember, when you have a body cavity, you have a space where the organ occurs. You have two sides to it. Remember, you took a balloon, you punch it in your fist, the balloon folded over. The surface closest to the organ is the visceral layer. The surface closest to the superficial part of the body, your skin, your epithelium, is the parietal layer. So serous membranes consist of two layers, your parietal layer and your visceral layer. Okay, there's three types of serous membranes. You have pleural, which lines the lungs. You have peritoneum, which lines your peritoneal cavity. And you have the pericardium, which lines the heart. Well, around the lungs, around the peritoneal cavity, and around the heart, you have a double membrane where the visceral layer is adjacent to or touching the organ, and the parietal layer is away from it toward the outside. Okay. So this is serous membrane lines your ventral cavities, peritoneal. One layer is peritoneal, one layer is pleural, and this is our connective tissue. Okay, and this is the serous, the exudate being excreted from it. Okay, cutaneous membrane. Cutaneous membrane makes up the skin. It consists of keratinized, stratified, squamous epithelium. Thick and waterproof. We added a new name. Stratify means what?
2.
Perfect. 2 or more layers. Squamous means what?
Flat.
Flat epithelium. Now keratinized. Keratin means there's a dead layer of cells over the top for protection. That's your keratinized layer. It's very thick and it's waterproof. So the skin is keratinized, stratified, squamous epithelium. Okay? So you can see your epithelium here. You can see your squamous cells that have nuclei right here. And as you get more towards the superficial surface here, towards the surface, you notice there's no nuclei. These are dead cells. This is keratinized tissue. So keratinized, stratified, squamous epithelium. Okay. Fourth membrane is your synovial membrane. These are fluid-filled membranes. They line joint cavities, your elbow, your knee, your temporomandibular joint. They produce synovial fluid that reduces friction within the joint. Okay? Can you imagine every time you talked, as you draw, there's the conda of the mandible rubs against the fossa, that bone was rubbing on bone, right? That would hurt a lot. So you produce synovial fluid. So there's fluid in between these two bones, your knee, your elbow, your hip, okay? As those move, the fluid cushions between those two joints and allow them to move freely. It's different than other membranes. There is no basal lamina or reticular lamina. It has gaps between cells because you want this fluid to flow now. You want the fluid to flow in between it to have gaps between the cells. Cells are derived from macrophages and fibroblasts. So it's a little different than the other membranes. Okay? Again, you have you have no basal lamina. It just is area surrounding it, this membrane, and no gaps, and you have gaps between the cell so the fluid can flow. And this is what it looks like. Okay? You have an articular cartilage, which is your hyaline cartilage. Again, it prevents bone-to-bone contact. You have synovial fluid that fills this joint here. This fluid fills by flowing between the cells and filling this joint. And in this capsule, this capsular layer that surrounds the bone and the synovial fluid and the hyaline cartilage, you have the outer layer of the capsule, you have capillaries, okay, where you can exchange oxygen, nutrients. You have adipocytes, which is for cushioning, and you have your areolar connective tissue. And the epithelium is adjacent to the synovial fluid. And that's how the joint's designed. Now the TMJ is an example of a synovial joint that can be moved. In fact, the temporomandibular joint, right in the jaw right here, is what a lot of times you see concussions on a football field, it's this joint being slammed into the skull. They did a study at the University of Texas about 5 years ago now, and they made special mouth guards for all the football players. And these football players wore those mouth guards during the game. There was not one concussion during that season. What causes most of the concussions is this jawbone being hit, and then the joint slams up into the skull, and that causes concussion to the brain. What the mouth guard is absorb that shock so that force cannot be transmitted through this joint. So that's good for football. Also, there was a study done, I forget who did the study, on weightlifters. When weightlifters wear a proper mouth guard, they allow the teeth to come together and don't clench in the joint, they can lift heavier weights. It allows a greater athletic performance. So a lot of athletes now are going to mouth guards. I make a lot of mouth guards for a lot of my friends that still racing motocross, and they wear them to protect the teeth, and they do very, very few concussions that we're seeing. So mouth guards do play a big reason for a lot of things. All right, connective tissue framework of the body. Those are your 4 types of membranes. Connective tissue creates the internal framework of your body. That's what holds everything together. That's around all your organs, around all your bones. That's your glue holding your body together. Layers of connective tissue connect organs with the rest of your body. Layers of connective tissue are called fascia. So when you hear someone cutting through fascia, a surgeon, they're cutting through layers of connective tissue. There's superficial fascia, there's deep fascia, and there's subserous fascia. If you had to take a guess, superficial fascia would be located where? Near the superficial part of your body, right? Okay, the deep one would be below that, subserous would be below that. I don't know if I hit the button or not. Let's see if this one advances a slide here. All right, so 3 different types of fascia. Went too far. All right, so here's your superficial fascia. Okay, it's between the skin and the underlying organ. So just below the skin is your superficial fascia. Okay, this is also known as the subcutaneous layer or the hypodermis. Everybody's heard of the hypodermic needle, right? Hypodermic needle delivers the drug or whatever is in the syringe to the superficial fascia. That's your hypodermis. That's what hypodermic was designed for. Okay. Everybody's heard of a sub-Q injection. Sub-Q is subcutaneous. That's the layer just below the skin, superficial fascia, where you're injecting your drug. That's a sub-Q injection. Deep fascia. Deep fascia forms a very strong fibrous network. It's dense connective tissue, okay, and it's below the superficial fascia. It's bound to your capsules, your tendons, your ligaments, your aponeuroses. It's all bound inside of there. Okay. Your subserous fascia is even deeper than that. It's below the deep fascia. It's between the serous membranes and the deep fascia. Okay. So superficial fascia is your hypodermic layer. Deep fascia is just deep to the superficial fascia, and subserous fascia is deep to the deep fascia. That's how it's set up. Muscle tissue. Okay, muscles have the ability to contract and relax. They have to have both. Okay? If you see a nursing exam and you have to pick what a muscle can do, it has to do 2 things. It has to contract and it has to relax. If it can't do either one of those, it's not going to function correctly. Okay? If you get contraction and it can't relax, you're in a world of hurt. Okay, there's 3 types of muscle cells. Okay? There's skeletal muscle, there's smooth muscle, and there's cardiac muscle. You need to know all those. Okay, the cells are different than typical cells. The cytoplasm is called sarcoplasm when it's inside a muscle cell. The plasmalemma is called the sarcolemma. The sarcolemma is just the membrane of a muscle cell. Same thing as a plasma membrane. A plasmalemma is a membrane of a regular cell. The sarcoplasm is the cytoplasm of a muscle cell. Okay, those are the only differences. Muscle cells are sometimes referred to as skeletal muscle fibers. They're multinucleated. Nuclei lie just under the sarcolemma. Okay, so you have multiple nuclei in one cell. They're incapable of cell reproduction. Okay, think about that for a second. If you have a trainer, you're going to the gym, and they tell you, we're going to break down your muscles so we can rebuild new ones. That is a physical impossibility. If they tell you they're going to break down your muscles, go to another trainer. You cannot reproduce muscle cells. You are born with as many muscle cells, skeletal muscle cells, as you're going to have. You're not going to make any more. Can you make them larger? Yes. Can you make them more effective, stronger? Yes. But you cannot ever produce more muscle cells. So if they're going to destroy your muscle cells, switch to a new trainer because you'll never regenerate them. Okay? That's why muscle injuries are so dangerous to track runners. Hamstring muscles, you pull a hamstring muscle, it never grows back. Now, you will bleed between the torn muscle, and that blood will form a scab, and that scab will hold the muscle together. but not near as strong as the normal muscle was. Okay, you cannot regenerate muscle cells. Okay, now myosatellite cells can reproduce, and therefore muscle repair is slightly possible. It's not great. You can get some muscle repair with myosatellite cells. Okay, but most of the time, if you damage a muscle, it's damaged for life, and it's cumulative. It keeps damaging. Okay, so have a striped appearance under the microscope, and they voluntarily move the skeleton. So skeletal muscles are under voluntary control. They cannot reproduce. They have multinuclei, and they cause a contraction and relaxation of muscles. Okay? And again, with myosatellite cells, you can get some repair is possible. Okay. Again, when you see the picture of them, this is what a skeletal muscle looks like. It's striated. Okay, you'll need to identify a histological slide of a skeletal muscle, a smooth muscle, and a cardiac muscle. You gotta be able to tell me the difference. I guarantee you'll see that on your midterm and on your practicum. Okay, there's multiple nuclei and there's striations. Everybody see the striations. These are little bands that come across, all these little bands here. If you see striations and you see nuclei lying in a line like this, you know you have a skeletal muscle, a histological sample of a skeletal muscle. Okay? No one's gonna miss that, right? Okay, so striations, long cells, skeletal muscles. Smooth muscles. Okay, smooth muscle is capable of cell reproduction. You can make more smooth muscles. They're at the base of your hair follicles. They're in the walls of your blood vessels, line the urinary bladder, respiratory circulatory system, digestive system, and reproductive tracts. Okay, they are capable of cell reproduction. Skeletal muscles are not. Your muscles that move your body. They have tapered ends. They're non-striated. So if you do not see striations, you have smooth muscles. They're for involuntary contraction. You cannot control them by thinking about it. Your body controls it. You don't control whether your blood vessel walls constrict or relax. Your body controls that. Your respiratory tract, okay, your hair follicles. You don't think about each hair follicle and whether it's going to form that muscle around it. Okay, it's involuntary. You cannot control it. Okay, again, that's your smooth muscle. Big difference. There's tapered ends. They're not long, and they do not have striations. Okay, and they're capable of reproduction. So here you go. Here's your histological sample. Again, notice here's your strands. They come to tapered ends from the end, and there are no striations, lines running up and down. Okay, everybody see that? No striations. They can tell the difference between this one and, okay, look at this slide right here. Remember what it looks like. Go back, right here. See the difference. This has striations. This has lines going up and down. This one does not. Okay, that's smooth muscle. Smooth muscle can regenerate. You can make more smooth cells, and there's no striations, and it is involuntary control. It moves food, urine, the reproductive tract, controls the diameter of your respiratory passages, regulates the diameter of blood vessels. Again, things you cannot control by thinking about. Cardiac muscle. So this, you had skeletal, excuse me, you had smooth, and now we got cardiac. Those are the three types. Cardiac muscle is found only associated with the heart, therefore the name cardiac muscle. Each cell has just one nucleus. Skeletal muscles has multiple nuclei. Cells are connected by intercalated disc. This is the key word. This is what's going to allow you to identify cardiac muscles on a histological slide. You will look for intercalated disc, and I guarantee you, the slide you have on your exam will show intercalated discs. Intercalated discs are there because the cells have to coordinate and all contract together. That's what these intercalated discs do. They allow the cells to communicate so the heart makes a effective pump, that everything contracts together. All right, so these are called pulsating contractions. The cardiac can also be called striated involuntary muscles. So cardiac muscle is striated, but it's going to have intercalated discs. That's what's going to determine the difference. And again, cardiac muscle is involuntary. The heart beats. You do not have to think about it. You do not have to tell your heart to beat. It's going to beat on its own contractions. So it's an involuntary muscle. Just like smooth muscle is involuntary, cardiac is involuntary. Smooth muscle does not have striations. Cardiac muscle does have striations along with intercalated disc. Here's your intercalated disc. This is a slide you're going to see. Okay, here's your striations. You can see the bands, not quite clear here, but you see bands going, and you see these lines that I'm tracing right here. These are intercalated discs. Once you see lines, deep lines like this, okay, like that, like this here, you know it's cardiac muscle, okay? So as you look on the left here, you can see the striations a little bit better, and you see these cracks or these deep lines. These are your intercalated discs. Once you intercalate disc, you got cardiac muscles. So your cells are short, they're branched, striated, usually single nucleus, and they're interconnected by intercalated discs. That's your key word. Okay? Cardiac muscle cells differ from skeletal muscle fibers in 3 major ways. Okay? Cardiac muscle cells are smaller. Cardiac muscle cells branch. And a typical cardiac muscle cell has one centrally placed nucleus. Both cardiac and skeletal muscles contain actin and myosin filaments that allow the contractions. And it both have striations. Okay, so you see striations in both. Cardiac ones be a little bit smaller, but the big key is intercalated disc. If you see intercalated disc, always either write down or circle cardiac muscle. Okay, so what are the 3 types of muscle? Skeletal, smooth, cardiac. Which one is involuntary? Smooth and cardiac. Which one is voluntary? Skeletal. Okay, skeletal ones are striated, easy to see. Smooth muscle is non-striated. Cardiac muscle is striated, but it has intercalated disc. That's how you're going to determine it for your exam. Does that make sense? Okay, neural tissue, nerve cells. Neural tissue is specialized to conduct electrical signals throughout your body. You have 2 types of neuronal cells. You have the neurons, which are the cells that actually transmit that impulse. They have that sodium potassium pump. They shoot that impulse down. The nerve fibers that says, hey, my hand's on a hot stove, move it. Okay, that's what interprets the actual pain or whatever signal or stimulus is being produced. Those are your neurons. Neuroglial cells are the supporting cells of the neurons. They protect the neurons and allow that transmission, that impulse, to travel extremely fast to the central nervous system. Okay, so neurons actually conduct the impulse, the electrical impulse. Neuroglial support it to make sure it stays healthy and make sure it can fire that impulse in a very fast and efficient manner. Okay, so again, your neurons transmit the impulses. Your neuroglial are your supporting cells. Okay, and they protect the neurons. Neurotissue, the neurotissue actually is neurons. That's what conducts the electrical impulse. They're the longest cell in the human body. Sometimes that's on your nursing exam, sometimes it's not. They'll say the longest cell in the human body is a neuron. It's incapable of cell reproduction. Okay, so just like cardiac cell, cardiac muscle cell, just like a skeletal muscle cell, the neural cell, the neuron, is incapable of cell reproduction. Now think about what that means. It means you're born with as many nerve cells as you're going to have. That's what makes up your brain. You're never going to produce another nerve cell, ever, your whole life. So if you damage a nerve cell, it stays damaged for life. If you go on a beer drinking binge, okay, it's going to kill nerve cells. That's what alcohol does. Okay? It kills neurons. In fact, one of the treatments of tic de la rue, which is trigeminal neuralgia we talked about, okay, that nerve gets triggered and you get that shooting pain. One of the treatments is to take alcohol, inject it into the nerve ganglion where those nerves are, and kill those nerves so that nerve can't transmit pain. So alcohol kills nerve cells. Now, not telling you not to go out and drink or anything like that, but when you go on these long binges, and it kills these nerve cells. That's it. They never regenerate, and it's cumulative. Every time you do it, it's more and more and more. And by the time you get old enough, those effects are going to become problems. Just like cardiac muscle, they never regenerate. So every damage that's going to a cardiac muscle, okay, gets cumulative. It never goes back. It's just additive to it. So again, neural tissue, the neurons, not the neuroglial cells, but the neurons are incapable of cell reproduction. They consist of the soma. Soma just means the cell body. They consist of an axon. An axon is the long part that comes out and it sends the nerve signal to another cell. And it consists of dendrites. Dendrites are sensory receptors of the cell where input comes in. And once the input gets big enough, that axon fires and transmits to the next cell. So you have soma, axon, and dendrite. Okay, and this is what the cell looks like. Okay, they're in the spinal cord, they're in your brain, they make up all your thought processes. So here's the soma. You can see my mouse moving here. It has a nucleolus and a nucleus of the soma. Okay, that's your cell body. All these little trees here, these little branches, these are dendrites. These receive signals from other axons, from whatever else it has, from other stimuli. These all receive signals. The soma vote on all these signals. Once the signal reaches the threshold level and they decide it's going to propagate that response, that it's a true response, then the impulse or the signal gets sent down this axon, okay, into either a target organ or other cells and gets turned along the line. So these are the three things you need to know. You need to know your dendrites, you need to know your soma, and you need to know the axon and how they work. Okay, and this is the light microscope. These are the dendrites again. This is a soma, and you see your axon starting to form right here, and it's going to go this direction. Okay, tissue nutrition and aging. Basically, after age 21, your body starts to break down, like it or not. After 21, things start to go downhill. Everything starts to break down, and it just gets worse and worse and worse. Repair and maintenance becomes less efficient as you age. Okay, I mean, I raced motorcycles, motocross for years. Okay, I raced it's been about almost 7 years now. I raced the Twin End Palms Grand Prix, and I went into a turn with a 16-year-old kid. We laid our bike sideways in the turn. I went high, he went low, and our handlebars hit, and they locked. And I went, if we go down, I'm going to break something. He's going to break something. He's going to miss 2 weeks of school. I'm going to miss 3 months of work. And I just pulled out of the turn, let him take the shot, and he went ahead. And I'm going, that's when I'm just going to ride street bikes, enjoy myself, and I don't need to do that anymore. Bones don't heal. Things don't heal when you get older as much as a younger kid. And younger kids are fearless. They do not care what they do. So I'll let them have the whole shot. All right, so hormonal changes, lifestyle changes also affect the function of tissue. Epithelial becomes thinner, connective tissue becomes more fragile. Cardiac muscles and neural tissues cannot regenerate, like we talked about. So minor damage adds up over time. If you have minor damage to your heart over 20 years, that's going to cause major problems. Minor damage to your brain, to nerve cells, over a 20-year span is going to cause problems. Things add up if you can't replace them. Okay, and as you get older, replacement of the things you can replace gets slower too. Okay, so in embryology, this is how they all develop. You get the formation of tissue, you have fertilization where the sperm and the egg combine, you have the zygote, which is the fertilized egg, then you start cellular division. So that one cell starts to divide and forms every single cell of the human body. You get the formation of what's called a blastocyst, which is a hollow ball of cells. You get formation of 2 layers of blastocyst. You get the trophoblast, which is going to form the placenta, which anchors into the uterus, which provides nutrition and oxygen and everything else for the developing fetus. And from the trophoblast, from the blastocyst, you also get the inner cell mass. So the inner cell mass forms your embryo. So once the blastocyst forms from the zygote, the fertilized egg, it breaks into 2 layers. One layer forms the placenta, the trophoblast. One layer forms the embryo, which is your inner cell mass. 3 germ layers form the 4 tissues of the body. Okay, so 3 of the germ layers formed in the embryo are going to form the 4 tissues of the body. Okay, So this is a representation of it. You see fertilization, zygote, okay? You get cell division, you get your blastocyst. The blastocyst forms your trophoblast, which is going to form the placenta. Okay? And then it forms the inner cell mass, which is going to form the embryo. Okay, development of epithelium. So you begin as simple epithelia, one layer. Everything begins that way. You form one layer first. It differentiates into functional epithelium and gland cells. Complex glands begin to form, endocrine and exocrine. Everybody remember the difference. What's endocrine? Releases it where?
Inside the body.
Perfect. Inside the body. Blood vessels, lymph vessels. Exocrine releases where?
Outside the body. Like sweat.
Outside the body. Perfect. Yep, sweat. Exactly right. Okay, so all epithelium begins as a simple epithelium. Okay, once you have that, then it becomes stratified and grows. Okay, grows as that. You get connective tissue forming underneath it. You get skin form. You get respiratory epithelium. You get all the different types of epithelium form from this one beginning simple layer. Then from this, you form the complex glands and the different duct systems and the different blood vessels that innervate the connective tissue underneath. Do these blood vessels innervate into the epithelial layers themselves? No. Epithelium does not have blood vessels. The connective tissue underneath it does. Okay. Again, with embryology, the endoderm forms your mesenchyme. Mesenchyme are your stem cells. Mesenchyme differentiates to form your chondroblast, your osteoblast, your blood, your lymph, your connective tissue, which is loose and dense. Chondroblasts form cartilage. Osteoblasts form bone. Blood forms red blood cells and white blood cells. Lymph forms your lymphatic cells, lymphocytes, and your defense system. Goes in your B cells and your T cells. And connective tissue forms your loose and dense connective tissue that holds everything together under the epithelium. Okay, and here's a diagram of it the same way, just we talked about, the mesenchyme, the osteoblasts, the osteocytes, your blood, your lymph, and so on. And that's how it forms. Now the organ systems develop within the embryo also. Again, we're just going over basic, you have a whole chapter on embryology, like 150 pages of it. So we'll get a lot more detail than this. This is just the overall introduction to it. So the ectoderm layer and the endoderm layer form, okay? At day 14, 2 weeks, cells migrate between those two layers, forming a mesoderm. At day 18, organs begin to develop. At day 28, you're at the beginning of all major organ systems. So at 28 days, 1 month, you've got a brain, you got a spinal cord, you got hands, you got feet, you got legs, you got all your major your organ systems, digestive system, nerve system, respiratory system, are all developing, starting at 1 month development. So that brings up the argument of, when does life start? You're not going to solve that here, I guarantee you. So many different organ systems they develop show similar patterns, okay, as they organize. Digestive system, respiratory, urinary, and reproductive system each include passageways, okay, that are lined with epithelium, surrounded by layers of smooth muscle. All of those start off exactly the same. Patterns are a result of the development process under the first 2 months of embryonic development. You can see how they form. A lot of our organ systems develop the same way, and then they start to differentiate. But they have the same epithelial systems, the same linings, the same processes. They have to create a lumen where things are resorbed, things are excreted, and so on.